Tonight's lecture concerns the cardiovascular system, specifically the heart, chapter 20 in your textbook, beginning on page 727. The heart contributes to homeostasis by pumping blood through blood vessels to the tissues of the body to deliver oxygen and nutrients and remove wastes. So the purpose of this chapter is to learn about the components of the cardiovascular system, focus on the anatomy and physiology of the heart, learn about the cardiac cycle, and discuss the various factors that influence heart rate and force of contraction. So here's a broad overview of the cardiovascular system. The essential components of the human cardiovascular system are the heart, blood, and the blood vessels. It includes the pulmonary circulation, a loop through the lungs where blood is oxygenated, and the systemic circulation, a loop through the rest of the body to provide oxygenated blood. Vessels carry blood through the circuits. So here's some vocabulary for you to learn. Arteries carry blood away from the heart. Capillaries, arteries branch into small passages called arterioles and then into capillaries. The capillaries merge to bring blood into the venous system and permit exchange between the blood and tissues. Veins. After their passage through the body tissue, capillaries merge once again into venules, which continue to merge into veins. Veins carry blood to the heart. And coronary vessels. The heart itself is supplied with oxygen and nutrients through a small loop of the systemic circulation. The heart. The heart is the pump responsible for maintaining adequate circulation of oxygenated blood around the vascular network of the body. The heart is a four-chamber pump, with the right side receiving deoxygenated blood from the body at low pressure and pumping it into the lungs. This is the pulmonary circulation, and the left side receiving oxygenated blood from the lungs and pumping it at high pressure around the body, and that's the systemic circulation. So this cartoon is on page 728, figure 20.1, and it shows the position of the heart and the associated structures of the mediastinum. The positions of the heart associated with the structures in the mediastinum are indicated by the dashed outlines. So here we're looking at a transverse plane, and you can see just posterior to the sternum there's a dashed line, and that's outlining the mediastinum. The heart is located in the mediastinum with 2/3 of its mass to the left of the midline. And this cartoon is the same figure, 20.1, on page 728. And here we're looking at an anterior view of the thoracic cavity. And again, you can see the mediastinum outlined with the dotted line. And then you can see how the various parts of the anatomy are associated with that. The heart pumps oxygenated blood to the body and deoxygenated blood to the lungs. In the human heart, there is one atrium and one ventricle for each circulation, and with both systemic and a pulmonary circulation, there are 4 chambers in total. The chambers are called the left atrium, the left ventricle, the right atrium, and the right ventricle. The coronary circulation system provides a blood supply to the heart muscle itself. The coronary circulation begins near the origin of the aorta by 2 arteries, the right coronary artery and the left coronary artery. After nourishing the heart muscle, blood returns through the coronary veins into the coronary sinus and from this one into the right atrium. The backflow of blood through its opening during atrial systole is prevented by the Thebesian valve. The smallest cardiac veins drain directly into the heart chambers. The pericardium. The pericardium is a double-walled sac containing the heart and the roots of the great vessels. The pericardial sac has two layers, a serous visceral layer and a fibrous parietal layer. It encloses the pericardial cavity, which contains pericardial fluid. The pericardium fixes the heart to the mediastinum, gives protection against infection, and provides the lubrication for the heart. So this is figure 20.2 on page 729. The pericardium is a triple-layered sac that surrounds and protects the heart. The heart is enclosed and held in place by the pericardium. So this cartoon is showing you a simplified relationship of the serous pericardium to the heart. Also, we can see the deeper serous pericardium, which is a thinner, more delicate mesothelial membrane that forms a double layer around the heart. The outer parietal layer of the serous pericardium lines the inside of the fibrous pericardium. The inner visceral layer of the serous pericardium, which is also called the epicardium, is one of the layers of the heart wall and adheres tightly to the surface of the heart. Between the parietal and visceral layers of the serous pericardium is a thin film of a few milliliters of lubricating serous fluid. This slippery secretion of the pericardial cells, known as pericardial fluid, reduces friction between the layers of the serous pericardium as the heart moves. The space that contains a few milliliters of pericardial fluid is called the pericardial cavity. The pericardium consists of an outer fibrous pericardium and an inner serous pericardium. The serous pericardium has two layers, the visceral and the parietal. The visceral and parietal layers are separated by the serous cavity, and that is a fluid-filled space. So here's the functions or the physiology of the pericardium. For one, it fixes the heart to the mediastinum and limits its motion. Secondly, it protects it from infections coming from other organs such as the lungs. It also prevents excessive dilation of the heart in cases of acute volume overload. And furthermore, it lubricates the heart. So this figure is 20.3. It's a variation of 20.3, I should say, and it's on page 731. It outlines the anatomy of the heart. So the heart consists of 4 chambers, 2 atria and 2 ventricles. The major blood vessels of the heart include the inferior and superior vena cava and the aorta and pulmonary artery, or we can also call that the trunk. So going back to page 729, figure 20.2, this shows the 3 layers of the heart wall. So you've got your epicardium and your myocardium and your endocardium. And based on your current level of anatomy vocabulary, you should be able to figure out which one is the most superficial and which one is the most deep layer according to the language. So the epicardium is composed of 2 tissue layers. The outermost is called the visceral layer of the serous pericardium. This is a thin, transparent outer layer of the heart wall that is composed of mesothelium. Beneath the mesothelium is a variable layer of delicate fibroelastic tissue and adipose tissue. The adipose tissue predominates and becomes thickest over the ventricular surfaces, where it houses the major coronary and cardiac vessels of the heart. The amount of fat varies from person to person, corresponds to the general extent of body fat in an individual, and typically increases with age. The epicardium imparts a smooth, slippery texture to the outermost surface of the heart. The epicardium contains blood vessels, lymphatics, and nerves that supply the myocardium. The middle layer, or the myocardium, is responsible for the pumping action of the heart and is composed of cardiac muscle tissue. It makes up approximately 95% of the heart wall. The muscle fibers, like those of the striated skeletal muscle, are wrapped and bundled with connective tissue sheaths composed of endomysium and paramysium. The cardiac muscle fibers are organized in bundles that swirl diagonally around the heart and generate strong pumping actions of the heart. Although it is striated like skeletal muscle, recall that cardiac muscle is involuntary like smooth muscle. The innermost layer, the endocardium, is a thin layer of endothelium overlying a thin layer of connective tissue. It provides a smooth lining for the chambers of the heart and covers the valves of the heart. The smooth endothelial lining minimizes the surface friction as blood passes through the heart. The endocardium is continuous with endothelial lining of the large blood vessels that are attached to the heart. So I'd like to talk a little bit more about the myocardium. It's a specialized form of muscle consisting of individual cells joined by electrical connections. The contraction of each cell is produced by a rise in intracellular calcium concentration, leading to the spontaneous depolarization, and as each cell is electrically connected to its neighbor, contraction of one cell leads to a wave of depolarization and contraction across the myocardium. So back to figure 20.3 on page 731. The chambers of the heart include 2 upper atria and 2 lower ventricles. The sulci, plural for sulcus, are grooves that contain blood vessels and fat and that mark the external boundaries between the various chambers. And this is figure 20.3 continued, but now we're on page 732. And this just shows the chambers of the heart, including the upper atria and the 2 lower ventricles, but now we're looking at a posterior view of it. Atria, thin-walled chambers that receive blood from the vena cava and the pulmonary veins. Ventricles, thick-walled chambers separated by the atria by the AV valves. Chordae tendineae, fibers attached to the AV valves. Papillary muscles, muscular projections on the inner wall of the ventricles. So this is figure 20.4 on page 733. Blood flows into the right atrium through the superior vena cava, inferior vena cava, and coronary sinus into the left atrium through 4 pulmonary valves. So this is the same figure on page 733, and we're looking in detail at the right atrium. The right atrium receives blood from the superior and inferior vena cava and the coronary sinus. Again, this is the same figure, but now we're looking at an anterior view of a partially sectioned heart. So this is actually from a cadaver. But we can see the right ventricle receives blood from the right atrium and sends the blood to the lungs. Same figure. The left atrium receives blood from the pulmonary veins. So this is also figure 20.4, but now this view is on page 734. The left atrium receives blood from the lungs and sends blood all over the body. The wall of the left ventricle is much thicker than that of the right ventricle. And looking at this transverse cross-section, that's pretty obvious. The heart has a fibrous skeleton. The fibrous skeleton of the heart forms the foundation for which the heart valves attach. serves as a point of insertion for cardiac muscle bundles, prevents overstretching of the heart valves, and acts as an electrical insulator. So this is figure 20.5 on page 735, and we can see that fibrous rings support the four valves of the heart that are fused to one another. Heart valves and circulation of blood. The valves of the heart open and close in response to pressure changes as the heart contracts and relaxes. The right and left atrioventricular valves prevent backflow of the ventricles into the atria, and the right and left semilunar valves prevent backflow from the arteries into the ventricles. So this cartoon is on page 736 and 737. It's figure 20.6. So the heart valves prevent the backflow of blood. That's the gist of them. On the top of the cartoon there, we can see what we call bicuspid valves because it has two flaps that open or close. On the bottom part of this figure, we see heart from a cadaver, and if we look there at the bottom right, we see the semilunar cusp of the aortic valve. So that's an example of a tricuspid valve. So this cartoon is also figure 20.6, but now we're looking specifically at page 737. And we can see that when one set of valves in the heart is open, the other set is closed. So there, with this superior view of the atria removed, the pulmonary and aortic valves closed, and the bicuspid and tricuspid valves are open. And then there at letter E, the superior view of the atria removed, pulmonary and aortic valves are open, and the bicuspid and tricuspid valves are closed. So moving on to figure 20.7 on page 738. This is the systemic and pulmonary circulations. So the left side of the heart pumps oxygenated blood into the systemic circulation to all tissues of the body except the pulmonary alveoli of the lungs. The right side of the heart pumps deoxygenated blood into the pulmonary circulation to the pulmonary alveoli of the lungs. So the heart pumps blood into two closed circuits with each beat. One is called the systemic circulation and the other is called pulmonary circulation. The two circuits are arranged in series. The output of one becomes the input of the other, as would happen if you attach two garden hoses together. The left side of the heart is the pump for the systemic circulation. It receives bright red oxygenated blood from the lungs. The left ventricle ejects blood into the aorta. From the aorta, the blood divides into separate streams, entering progressively smaller systemic arteries that carry it to all organs throughout the body, except for the pulmonary alveoli of the lungs, because though the pulmonary alveoli is supplied by the pulmonary circulation, which we'll talk about in a minute. In systemic tissues, arteries give rise to smaller diameter arterioles, which finally lead into extensive beds of systemic capillaries. Here, exchange of nutrients and gases across thin capillary walls occurs. Blood unloads oxygen and picks up carbon dioxide. In most cases, blood flows through only one capillary and then enters a systemic venule. Venules carry deoxygenated blood away from tissues and merges to form the larger systemic veins. Ultimately, the blood flows back to the right atrium. The right side of the heart is the pump for the pulmonary circulation. It receives all of the dark red deoxygenated blood returning from the systemic circulation. Blood ejected from the right ventricle flows into the pulmonary trunk, which branches into pulmonary arteries that carry blood to the right and left lungs. In pulmonary capillaries, around pulmonary alveoli, blood unloads carbon dioxide, which is exhaled, and picks up oxygen from inhaled air. The freshly oxygenated blood flows into pulmonary veins and returns to the left atrium. Coronary circulation begins on page 738, but blood flow through the coronary arteries delivers oxygenated blood and nutrients to the myocardium. Its branches arise from the ascending aorta. Coronary veins remove carbon dioxide and waste from the myocardium, and those branches converge at the coronary sinus. And this cadaver heart is actually on page 740, figure 20.8. These cartoons are also part of figure 20.8, page 740. The left and right coronary arteries deliver blood to the heart. The coronary veins drain blood from the heart into the coronary sinus. So in this cartoon of coronary circulation, the views of the heart from the anterior aspect, letter A, and in B, it is drawn as if the heart were transparent to reveal blood vessels on the posterior aspect. So the cardiac muscle cells are self-excitable and therefore autorhythmic. Cardiac muscle cells repeatedly generate spontaneous action potentials that trigger the heart contractions. These cells form the conduction system, which is the route for propagating action potentials through heart muscle. And this is figure 20.10 on page 745. The conduction system ensures that the chambers of the heart contract in a coordinated manner. This is also figure 20.10 on page 745. Autorhythmic fibers in the SA node, located in the right atrial wall, act as the heart's pacemaker, initiating cardiac action potentials that cause the contraction of the heart's chambers. So this figure goes back to page 743, figure 20.9, and this just shows cardiac fiber muscles connecting to neighboring fibers by intercalated discs, which contains desmosomes and gap junctions. So we should remember desmosomes and gap junctions from our muscular system discussion. And remember that the intercalated discs are unique to the cardiac muscle. And now we're back at page 745, figure 20.10. And we see the conduction system that ensures the chambers of the heart contract in a coordinated manner. So we've got these two things called nodes that are there in the right atrium. The SA node, or the sinuatrial node, is located in the right atrial wall, and it acts as the heart's pacemaker, initiating cardiac potentials. And it also helps to cause the contraction of the heart's chambers. The AV node, or the atrioventricular node, is a structure that connects the electrical systems of the atria and the ventricles. And this provides some electrical impedance from the atria. This depolarization and contraction of the heart is controlled by a specialized group of cells localized in the sinoatrial node and the right atrium, in other words, the pacemaker cells. These cells generate a rhythmical depolarization which then spreads out over the atria to the atrioventricular node. The atria then contract, and this pushes blood into the ventricles. The electrical conduction passes via the atrioventricular node to the bundle of His, which divides into the right and left branches and then spreads out from the base of the ventricles across the myocardium. This leads to a quote-unquote bottom-up contraction of the ventricles, forcing blood up and out into the pulmonary artery and the aorta. The atria then refill as the myocardium relaxes. Influences on the conduction system. The autorhythmic fibers of the SA node are the natural pacemaker of the heart because they initiate action potentials most often. Signals from the nervous system and hormones like epinephrine can modify the heart rate and force of contraction, but they do not set the fundamental rhythm. So this is figure 20.11 on page 746. And this is action potential in a ventricular fiber. So an action potential in a ventricular an action potential in a ventricular contractile fiber is characterized by a rapid depolarization, plateau, and repolarization. So the first step is rapid depolarization due to sodium ion inflow when voltage-gated fast sodium ion channels open. 2, we have the plateau stage, and this is due to the calcium ion inflow when voltage-gated slow calcium ion channels open, and potassium ion outflow when some potassium ion channels open. And then finally, 3, we get repolarization due to the closure of the calcium ion channels and the potassium ion outflow when additional voltage-gated potassium ion channels open. So this is figure 20.12 on page 747. And this is an example of an electrocardiogram. We can call this an EKG or an ECG. But an EKG is a recording of the electrical changes that accompany each heartbeat. So an EKG is a composite record of action potentials produced by all of the heart muscle fibers during each heartbeat. The instrument is used to record changes in an electrocardiograph. And the image that we're looking at is just an example of an electrocardiograph. So here we see figure 20.13 on page 749. So this is the timing and route of action potential depolarization and repolarization through the conduction system and myocardium. Green indicates depolarization, and red indicates repolarization. So depolarization causes contraction, and repolarization causes the relaxation of cardiac muscle fibers. Good quiz or exam question. So at step 1, the depolarization of the atrial contractile fibers produces a P wave. And then at step 2, we get atrial systole contraction. At step 3, we get the depolarization of the ventricular contractile fibers producing a QRS complex. 4, the ventricular systole contraction. 5, the repolarization of the ventricle contraction fibers produces what we call a T wave. And then 6, we get the ventricle diastole, and that's the relaxation of the heart. The cardiac cycle. And this is page 748. The period between the start of one heartbeat and the beginning of a next heartbeat is called the cardiac cycle. During a cardiac cycle, each heart chamber goes through systole and diastole. In other words, the squeeze is called systole and normally lasts for about 250 milliseconds. The relaxation period, when the atria and ventricles refill, is called diastole. The time given for the diastole depends on the heart rate. One cardiac cycle consists of the contraction, systole, and the relaxation, diastole, of both atria, rapidly followed by systole and diastole of both ventricles. This includes electrical events, pressure changes, heart sounds, volume changes, and mechanical events. So this figure spans the next several slides, and this is figure 20.14 on page 750. But we can see we've got our EKG diagram, and at step 1, a depolarization of the SA node causes atrial depolarization marked by the P wave in the EKG. And then at step 4 there, letter Q, the QRS complex in the EKG marks the onset of ventricular depolarization. And then at number 8, the T wave in the EKG marks the onset of ventricular repolarization. Moving on to the next slide. So dealing with pressure changes, at number 5, ventricular depolarization causes ventricular systole. As ventricular systole begins, pressure rises inside the ventricles and pushes blood up against the AV valves, forcing them shut. For about 0.05 seconds, both the semilunar and AV valves are closed. This is the period of isovolumetric contraction. During this interval, cardiac muscle fibers are contracting and exerting force but are not yet shortening. Thus, the muscle contraction is isometric. Moreover, because all 4 valves are closed, ventricular volume remains the same, hence the term isovolumetric. This next slide shows heart sounds. And moving on to the next slide, volume changes. So at number 3, atrial systole contributes to the final 25 milliliters of blood to the volume already in each ventricle. The end of the atrial systole is also the end of the ventricular diastole. Thus, each ventricle contains about 130 milliliters at the end of its relaxation period. This blood volume is called the end diastolic volume, or EDV. And then at number 7, the left ventricle ejects about 70 milliliters of blood into the aorta, and the right ventricle ejects about the same amount of volume of blood into the pulmonary trunk. The volume remaining in each ventricle at the end of the systole is about 60 milliliters. The volume ejected per beat from each ventricle equals the end diastolic volume minus the end systolic volume. So that would be SV equals EDV minus ESV. At rest, the stroke volume is about 130 milliliters minus 60 milliliters equals 70 milliliters, a little more than 2 ounces. And then this is at the very bottom of the slide, and we can see that these are mechanical events associated with the heart. And this slide just kind of shows the whole figure 20.14 as 1, as you would see it on your page in the book. Cardiac output is the volume of blood ejected from the left or right ventricle into the aorta or the pulmonary trunk each minute. Stroke volume is the amount of blood pumped out of the ventricle in one beat. So the cardiac output equals the stroke volume times the heart rate. So there's a formula for you to review. There are 3 factors that regulate stroke volume: preload, contractility, and afterload. So preload is the degree of stretch on the heart before it contracts. Contractility is the forcefulness of contraction of an individual ventricular muscle fiber. And then afterload is the pressure that must be exceeded before ejection of blood from the ventricles can occur. Regulation of heart rate. Several factors regulate heart rate. These factors are the autonomic nervous system, hormones, ions, age, gender, physical fitness, and temperature. Heart rate regulation continued. Autonomic innervation. So again, there's a typo there. Autonomic is spelled wrong. But this heart rate regulation deals with the parasympathetic branch, specifically the vagus nerve. And there is also some interaction between the sympathetic branch of the nervous system. Hormones also can affect it. Epinephrine, norepinephrine, thyroid hormone, and the venous return also affects that. So again, there's another typo there. Return is spelled wrong. Apologies for that. Like I said, I get these from the publisher, and that's how I got it. So I apologize for any typos on here. So moving on to figure 20.16 on page 754. The cardiovascular center is in the medulla oblongata, and it controls both sympathetic, which is in the blue, and parasympathetic nerves, which are in the red, and those innervate the heart. So we can see that the input to the cardiovascular center from higher brain centers and from sensory receptors. So as far as the sensory side of it goes, your environment can affect your heart rate. Like if you get frightened, your heart rate may go up. And then on the parasympathetic side, we see the increased rate of spontaneous depolarization in the SA node and the AV node increases the heart rate. Increased contractility of the atria and ventricles increases stroke volume. And then the vagus nerve, which is on the parasympathetic side of it, deals with the decreased rate of spontaneous depolarization in the SA node, and the AV node decreases the heart rate. So here we see figure 20.17 on page 756. And I'm going to let you go through this slide on your own time. This deals with factors that increase the cardiac output. Moving on to exercise in the heart, regular aerobic exercise can increase cardiac output, increase HDL, decrease triglycerides, improve lung function, decrease blood pressure, and assist in weight control. So this is figure 20.18 on page 757. And we're going to talk about helping failing hearts. So cardiac transplantation is the replacement of a severely damaged heart with a normal heart or from a brain dead or recently deceased donor. So this is table 20.1 beginning on page 758. So this talks about cardiac assist devices and procedures. And as I usually do, I'm going to let you go through this table on your own time. So please make sure you read through this table. And this slide here is a continuation of table 20.1. And same thing for this slide. This is also part of 20.1, so please make sure you read through this. So moving on to the development of the heart, this is figure 20.19 on page 759. The heart begins its development from a group of mesodermal cells called the cardiogenic mesenchyme during the 3rd week after fertilization. Here we have more development of the heart. This is figure 20.20 on page 760. Partitioning of the heart begins on the 28th day after fertilization. So what that means is that the chambers of the heart begin to form about 28 days after fertilization. Moving on to disorders, homeostatic imbalances. This includes coronary artery disease, arteriosclerotic plaques, congenital heart defects, arrhythmia, and congestive heart failure. A heart attack occurs when the blood flow that brings oxygen to the heart muscle is severely reduced or stopped. This happens because coronary arteries that supply the heart with blood can slowly become thicker and harder from a buildup of fat, cholesterol, and other substances called plaque. This slow process is known as arteriosclerosis. If the plaque breaks open and a blood clot forms, that blocks the blood flow, and then a heart attack will occur. Collateral circulation is a network of tiny blood vessels which under normal conditions is not opened. When the coronary arteries narrow to the point that blood flow to the heart muscle is limited, collateral vessels may enlarge and become active. This allows blood flow around the blocked artery to another artery nearby or to the same artery past the blockage, protecting the heart tissue from injury. Moving on to the next slide, congenital heart disease. Congenital heart defects are problems with the heart structure that are present at birth. In some cases, doctors can find these problems during pregnancy. Many of these defects are simple conditions. They need no treatment or are easily fixed. Some babies are born with complex congenital heart defects. These defects require special medical care soon after birth. Things that make them more likely include problems with genes or chromosomes in the child, such as Down syndrome, taking certain medications, alcohol or drug abuse during pregnancy, viral infections like rubella in the mother during the first trimester of pregnancy. Types. Most congenital heart problems are structural issues like holes and leaky valves. Congenital heart defects are problems with the heart structure that are present at birth. These defects can involve the interior walls of the heart, the valves inside the heart, the arteries and veins that carry blood to the heart or body. Congenital heart defects change the normal flow of blood through the heart. There are many types of congenital heart defects. They range from simple defects with no symptoms to complex with severe symptoms, life-threatening symptoms. Heart valve defects. One may be too narrow or completely closed. That makes it hard for blood flow to get through. Sometimes it can't get through at all. In other cases, the valve might not close properly, so the blood leaks backward. Problems with the heart's walls. Well, it could be the ones between the chambers, the atria and the ventricles of your heart. Holes or passageway between the left and right side of the heart might cause blood to mix when it shouldn't. Issues with the heart's muscles. These can lead to heart failure, which means the heart doesn't pump as efficiently as it should. And then bad connections among blood vessels. In babies, this may let blood that should go to the lungs go to other parts of the body instead, or vice versa. These defects can deprive blood of oxygen and lead to organ failure. Examples of simple congenital heart defects. Holes in the heart, or septal defects. The septum is the wall that separates the chambers on the left and right sides of the heart. The wall prevents the blood from mixing from the two sides of the heart. Some babies are born with holes in the septum. These holes allow the blood to mix between the two sides of the heart. Atrial septal defect, or ASD. An ASD is a hole in part of the septum that separates the atria, the upper chambers of the heart. The hole allows oxygen-rich blood from the left atrium to flow into the right atrium instead of flowing to the left ventricle as it should. Many children who have ASDs have few, if any, symptoms. Ventricular septal defect, or VSD, is a hole in the part of the septum that separates the ventricles, the lower chambers of the heart. The hole allows oxygen-rich blood to flow from the left ventricle into the right ventricle instead of flowing into the aorta and out of the body as it should. So here we see a cross-section of a normal heart and a heart with ventricular septal defect. Patent ductus arteriosus. PDA is a fairly common heart defect that can occur soon after birth. In PDA, abnormal blood flow occurs between the aorta and the pulmonary artery. Before birth, these arteries are connected by a blood vessel called the ductus arteriosus. This blood vessel is an essential part of fetal blood circulation. Within minutes or up to a few days after birth, the ductus arteriosus closes. In some babies, however, the ductus arteriosus remains open. The opening allows oxygen-rich blood from the aorta to mix with oxygen-poor blood from the pulmonary artery. This can strain the heart and increase blood pressure in the lung arteries. Narrowed valves. Simple congenital heart defects also involve the heart's valves. These valves control the flow of blood from the atria to the ventricles and from the ventricles into the two large arteries connected to the heart, the aorta and the pulmonary artery. Valves can have the following types of defects. Stenosis. This defect occurs if the flaps of a valve thicken, stiffen, or fuse together. As a result, the valve cannot fully open. Thus, the heart has to work harder to pump blood through the valve. Atresia. This effect occurs if a valve doesn't form correctly and lacks a hole for the blood to pass through. Atresia of a valve generally results in more complex congenital heart diseases. Regurgitation. This defect occurs if a valve doesn't close tightly. As a result, blood leaks back through the valve. The most common valve defect is pulmonary valve stenosis, which is a narrowing of the pulmonary valve. This valve allows blood to flow from the right ventricle into the pulmonary artery. The blood then travels to the lungs to pick up oxygen. So here's some example of complex congenital heart defects. Complex congenital heart defects need to be repaired with surgery. Advances in treatment now allow doctors to successfully repair even very complex congenital heart defects. The most common complex heart defect is tetralogy of Fallot, which is a combination of 4 defects. Those 4 defects are pulmonary valve stenosis, a large VSD, an overriding aorta, and right ventricular hypertrophy. And that concludes tonight's lecture.